Okay, so now I will proceed to subunit 1I. Okay, so this is a discussion on GNP in various formulations. So the drugs formulation comes in various different shape, different formulation, different types. So each of these might have different aspects in terms of the GMP. So we will discuss four types of formulation here. One is sterile products, and then radiopharmaceuticals. The third one will be in herbal products, and lastly, GMP in liquids, creams, and also ointments. So the first types of formulation is sterile product.
Sir,
Yeah.
slide kita orang tak nampak.
Oh, okay, okay. Oh, sorry, saya tak share. Sorry, Okay, sorry, I repeat again. The outline for the subunit 1I, it will be on GMP for sterile products, GMP in radiopharmaceutical. The third one will be GMP in herbal products. And lastly, GMP in liquids, creams, and also ointment. So the first one is sterile product. So here the image is what you see. It's like an ampule over here. You have an ampule where the cap is, I mean the lid are tightly sealed. Okay, once open, then you cannot. I mean it has to be finished. You cannot keep it and then you want to use it on a day. So this is what it means by sterile. So sterile products, products that are going to be administered using an enteral route of administration, which are going to be infused directly into the bloodstream or body tissue. So I think you are already familiar with the types of formulation because these are, I think, covered in pharmaceutics. We have okay, so the specific requirements in sterile products you because it has the certain special need as in order to minimize risk of microbiological contamination and also pyrogen contamination. So the production should be carried out in clean areas. So it cannot be in the same place as you want to manufacture or produce the usual tablet, for example. This has to be done in clean areas, or we might also call it as clean room. And then only minimum number of personnel required should be present in clean areas, so cannot be crowded. So maybe 1 or 2%, 3%, okay, or just the specific staff, okay, that is intended to work in that clean area should be present. Okay, otherwise it is not advisable to have other personnel there as well. And then inspection and controls conducted outside of clean areas. And all personnel should receive regular training, includes the most important one is hygiene and also basic elements of microbiology. So why you need to know about microbiology? So that you know the things you have to avoid, okay, to uh, have a microbiological uh, uh, contamination, okay, like this one. And then the staff that are working inside the clean areas have to avoid from wearing wristwatches, makeup, and also jewelry. So all of these are normally avoided. In fact, jewelry, even in normal production areas, jewelry are also, I think, prohibited because there are a lot of moving machine. Okay, so if let's say you wear a ring, for example, and the ring accidentally got or removed from your finger, so it might got stuck into one of the machine, and this can lead to problem. And you should follow the clothing requirement based on the clean areas. So in clean areas, this is the usual attire. We call it as a smoke suit. Okay, but this one got like a breathing apparatus as well, attached to the waist over here. But certain places might not require this. Okay, depends. But if this type of smoke suit, if they require this breathing apparatus, that means the suit itself is tightly, tightly sealed lah. Okay, so that's why they have this sort of like breathing apparatus attached. And then the clean areas is not actually just for sterile product. I mean, in terms of pharmaceutical industry, but for semiconductor manufacturing, okay, that produce microchip. Okay, companies like what is it? Texas Instrument, Infineon. This is companies in Malaysia where they produce these microchips and everything. They also have to be produced in the clean room, okay, or clean areas. So all of the staff that needs to go inside the production areas have to wear similar attire, because when we talk about microchip, they are very small sizes, and you cannot have dust on them. Okay, they might not have the microbiological contamination, but we don't want to have all of this dust in the production line. So the requirement for premises in terms of clean areas, they are graded with A, B, C, and D. So grade A clean areas are zoned for high risk operation, and it has to have laminar airflow workstation. Laminar airflow is similar to what do you call it, a fume hood, okay, where the air is being sucked out of the cabinet, okay, for example. And then B for aseptic preparation and filling, grade C and D to carry out less critical stages in production of sterile products. So all exposed surface should be smooth and unbroken, so that it will minimize particles and microorganism contamination. So sinks and drains are prohibited at grade A and B. So these two areas, there shouldn't be any sink and also drain. And the changing room for personnel that wants to go in and out of the clean room must have an airlock. Okay, so it must have an airlock. And then a warning system should also be provided to indicate failure in air supply. Okay, so there must be a warning system, or before the warning system, the air indicator supply, okay, also must be present. And then for equipment, maintenance and repairs should be carried out outside the clean areas. So normally they are taken out from the clean areas, and then only they will conduct the maintenance and repairs for any equipment. Water treatment plants and distribution system should be designed, constructed, and also maintained. And just make sure that they also is not operated beyond their capacity. And water for injection should be produced, stored, and distributed in a manner which prevent microbial growth. So the temperature advisable is above 70 degrees Celsius. And then the QC for the sterile products, the sterility test applied for all of the finished product. Okay, you have to make sure that the finished product for the sterile products must pass the sterility test. And then special attention also must be paid to the validation and the monitoring of the entire manufacturing process. the sterilization process, okay, the GMP for the sterilization process, it should be validated, and then biological indicators also has to be considered as an additional method for monitoring the sterilization. That means this is in terms of microbiological growth. If you see any contamination, so that indicates fail in terms of sterilization, and the records must be available and approved as part of the batch release procedure. That means the sterilization record must be included as one of the document or requirement needed, okay, for that batch to be released. Okay, the second type of formulation is radio or radiopharmaceuticals. So radiopharmaceutical means they are what do you call that, biohazard or reactive, bioreactive. So radiopharmaceutical, so this is the definition. It is used either for diagnostic or therapeutic purpose, composed of a radioisotope bond to an organic molecule. So diagnostic is like they act as a dye, not DIE, DYE, dye, or like a coloring agent. Okay, a patient have to consume and maybe underwent a CT scan, for example, or a PET scan. Then the radiopharmaceutical dye will interact maybe with cancer cell, for example. So when you do the scan, the area which is affected, by the cancer cell will have a different colors. Okay, so that is for diagnostic lah. But for therapeutic, mostly they are used to treat cancer as well. And then manufacturing and also handling, because they are potentially hazardous. Okay, they might, because they contain this radioisotope, so they might be radioactive. And due to the short shelf life, products may be released before completion of all QC tests. So even before the test has been finished, the products are maybe on the way to the customer or distributor. In Malaysia, I know one place that produce this radiopharmaceutical, which is in Agensi Nuklear Negara, because they have the nuclear reactor over there. Okay, even a small one, that nuclear reactor can help. It is able to produce this diagnostic radiopharmaceutical product to be used in terms of radiography. Okay, and the personnel that involved in radiopharmaceutical should have additional competence in radiation protection, training in radiopharmaceutical specific aspects. So this is very, very important in terms of the radiation protection, because if let's say the personal protective equipment, the PPE specific for radiation are not properly wear, and the person works there every day, maybe for a longer period, they might also be exposed, okay, to the harmful radiation. Okay, end up the person who works there might get cancer. And on the premises and also equipment, the important measures should be established and implemented to prevent cross-contamination from personnel, materials, and radionuclide. Closed or contained equipment should be used whenever appropriate, and then precautions should be taken to avoid radioactive contamination within the facility. And the radioactive should be able to be detected using a detector, and also by routine testing or routine swab testing. The next GMP aspect is the QC, okay, quality control. So before testing is performed, sample can be stored to allow sufficient radioactivity decay. All of the tests, including sterility tests, should be performed as soon as possible. So you cannot delay. and returned products may not be reprocessed and must be stored as radioactive waste. So if let's say the customers suddenly wants to return an extra formulation, for example, radio pharmaceutical product, so you cannot just put it in your stock inventory. Okay, it has to be straight away be considered as radioactive waste. Then on reference and retention sample, sufficient samples of each batch of bulk should be retained for at least 6 months after expiry of the finished product. So even after the expiration date, you have to keep at least another 6 months. Okay, and then starting material should be retained for at least 2 years after product release. So these are the timeline for sample retention. Okay, the third type of formulation is a herbal product. So GMP herbal product. So you see here all of these capsule, but each of the capsule are in different color. So there could be different types of herbal product here. The yellow one might be from turmeric, for example. Okay. So what can be considered as herbal product? It can be a medicinal plant, an herbal substance, or any herbal preparation. So because of their often complex and also variable nature, control of starting materials, storage, and processing assume particular importance in the manufacture of herbal medicinal products. So the first aspect, which is the premises, herbal substance must be stored in separate areas. Okay, they must have a specific area for it. Normally, the starting material, maybe if you have dried plants, they are kept in a herbarium, for example, herbarium. And then storage areas have protection against the entry of insects or other animals such as rodents. So you have to make sure that it is what they call that, insect proof. So no insect or what they call that, pest can be seen entering the storage area. Okay, not even an ant. Okay, semut pun tak boleh. The production area should have provision during process whenever dust is generated to facilitate cleaning and to avoid cross-contamination. So maybe there is an exhaust fan to suck out of any dust generated from the process, for example. Okay, if your starting material is from leaf of a plant, so after drying and then you want to grind all of the leaves, so this might cause dust lah. Okay, so you have to consider this as well in terms of the premise area. And then the equipment that used must be compatible with the extraction solvent in order to prevent any release or undesirable absorption of substance that could affect the product. So here it is more or less want to say that the equipment that you are using should be compatible with your extraction method. Okay, so here what you see in the image is a very large extraction vessel. Okay, in our lab normally you will do it in a, what do you call that, separating funnel, for example, or but here this is large scale. And the QC for herbal product, due to the fact that medicinal plant or the herbal substances are heterogeneous in nature, their sampling should be carried out with special care by personnel with particular expertise. A reference sample of the plant material is necessary. So normally when you want to make sure that the plants that you have are actually the same, it comes from the same tree or the same species, you would require a botanist. Okay, that is a person who I would say an expert in terms of plants. We call them a botanist, and these botanists have the ability, I mean, they also have the authority to identify the plant and also species of the plants. So QC personnel should have particular expertise and experience in herbal substance, herbal preparation, and also herbal medicinal products in order to be able to carry out identification tests and recognize adulteration, the presence of fungal growth, and also infestation. So here are the processing instruction in terms of the herbal products. It should describe the different operation carried out upon the herbal substance, such as cleaning. So here in the image, you see leaf, okay, leaf of a plant, I think. So it includes cleaning, drying, crushing, and sifting, and also include drying time and what is the temperatures and method that are being used to control cut size or particle size. So these are all has to be detailed out, okay? And then the second criteria, whereby you should describe the cleaning and selection of plant material before storage. And you should include details of solvent, time and temperatures of extraction, and details of any concentration, stages, and also methods used. Okay, the last formulation that we will discuss is liquids, creams, and also ointment. So here in image you see an eye wash. So eye wash is liquid, and then you have a emollient cream here, and the last one is an ointment. So creams are actually water-based medication for topical use, while ointments are oil-based medication for topical use. And liquids, practically just water-based, or aqueous. Okay, so liquid creams and ointments may be particularly susceptible to microbial and also other contamination during manufacturing. Therefore, special measures must be taken to prevent any contamination. So on the premises and also the equipment, it is recommended to use closed system of processing and also transfer to avoid contamination. And equipment must be designed that include a minimum of sites where residues can accumulate and also promote microbial proliferation. And the use of glass apparatus should be avoided wherever possible, because we want to use everything in stainless steel. So high quality stainless steel is often the material of choice for product contact parts. And during the production process, water used in production should be specified and monitored. And it is best to use the ultra pure water supply instead of just taking from tap water and then you just do a normal filtration. Okay, normally we use very high high purity of water lah, void of any contamination. And then you should take extra care when transferring materials via the pipelines to ensure that they delivered to their correct destination. So all of the pipelines that you see in the image here, I mean, you have to make sure that they are connected as what you intended, okay, so that they flow correctly. And during the production, materials likely to shed fibers or other contaminants like cardboard or wooden pallets should not enter the areas where products or clean containers are exposed. And then during the filling process, the mixtures that are being filled into the container, you have to make sure that it is homogeneous. Okay, I mean, like this, the color of the cream or this ointment is orange, so all of it should be orange. Okay, not on the bottom is orange and suddenly up until the top over here it becomes slightly yellowish. So it has to be homogeneous. Okay, so that is the end for topic 1H, which is discussing on the